Happy Monday. Welcome to the last week of first trimester. Just a couple of things that we're going to talk about today, and then you're going to have an activity that you're working on for the rest of the time. So first, take a minute and review some of the things that you were reading about in the graphic novel that you hopefully finished up by last night. So when life first originated on Earth and we formed those earliest types of biomolecules and those really simple protocells, the earliest types of cells were prokaryotic. Prokaryotic cells are really simple. They don't have a lot of different parts and different structures. They do have a cell membrane. They do have genetic material, but it's not enclosed in a nucleus. They have ribosomes to make proteins, but they don't have a lot of the other more complicated structures that we find in eukaryotic cells. So what you read about in that graphic novel was kind of how some of the first eukaryotic cells may have originated. And part of that process is a theory called endosymbiosis, which is actually kind of interesting. The theory here is that some early prokaryotic cell at one point early in the history of living things ingested, it surrounded another prokaryotic cell. So at this point, these are both prokaryotes. One's just bigger than the other. And some prokaryotes would ingest and digest other cells, and that's how they got their food. But in this case, they happened to ingest one that was an aerobic bacteria. Aerobic means it can use oxygen. In other words, it can perform some process similar to cellular respiration as we know it today. And so it ingested this bacteria, but instead of digesting it and breaking it down, those bacteria formed a symbiotic relationship and lived inside of this other prokaryote. It turned out to be a really good arrangement because these little aerobic bacteria provided oxygen and energy. Sorry, they provided energy. They used oxygen. They provided energy to this larger prokaryotic cell. And they got this sheltered place and they got the nutrients and things that they needed to live and survive. So they weren't eaten by anything else. So those early aerobic bacteria that were ingested first, over time, evolved in such a way that they became dependent on that host cell and they couldn't live on their own anymore. And they evolved into the organelles that we have in our cells that use oxygen to produce energy. They became the mitochondria. We know these came first because all cells, both plant cells and animal cells, have mitochondria. So we know those aerobic bacteria had to be ingested first. This process then happened again with another type of bacteria called cyanobacteria that were able to photosynthesize. They could take sunlight and use it to produce sugars like glucose. Those were also ingested and became another symbiotic bacteria that eventually couldn't live on its own. Those became chloroplasts. And again, we know that one must have happened second because not all eukaryotic cells have chloroplasts, but they all have mitochondria. A couple pieces of evidence that help support this theory. First of all, if you just look at the size, mitochondria and chloroplasts are about the same size as a lot of other prokaryotic cells that exist today. And so that is evidence that maybe they were prokaryotes at one point. Mitochondria and chloroplasts also contain little bits of their own genetic material. They actually have their own DNA, and they are able to replicate themselves within your cells. Mitochondria can copy themselves if your cell needs to produce more energy. And so they can replicate themselves just like a prokaryotic cell would be able to do. The other thing that provides some evidence is both mitochondria and chloroplasts are surrounded by a double membrane. And we'll look at that a little more closely when we study energy. But you can see here the way that that double membrane around those organelles evolved is one of the membranes, the inner one, was actually the membrane of that original prokaryote. The second membrane was this kind of bubble of the host cell membrane that formed as it took that bacteria in. It's kind of like a little vesicle or a vacuole within the cell. And so you have two layers of membrane, and these organelles, the mitochondria and chloroplasts, still have two layers of membrane around them. So there's quite a bit of evidence that supports this theory of endosymbiosis as the origin for some of the organelles found in eukaryotic cells. Last week, you did an experiment looking at surface area and volume, and how that ratio is affected by things like cell size and shape. You probably found that, well, you probably noticed that the higher the surface area to volume ratio, so that's the bottom line of numbers down here, the higher that ratio, the faster diffusion happened into those cells. So if you cut several blocks that were like cubes of different sizes, the littlest one probably turned yellow first. It had the highest surface area to volume ratio. It was a real quick thing for the vinegar to diffuse all the way in there. This ratio is affected not only by the size, but also the shape. Like I just said, smaller cells tend to be more efficient. This is one of the reasons that cells are very small. This also means that prokaryotic cells, since they are smaller, have a higher ratio and are more efficient than eukaryotic cells. We'll come back to that idea in a minute. This is also affected by the shape. If you cut any that were really long and thin, you probably noticed they also turned yellow sooner than some of the other cells. And so certain types of cells have particular shapes in order to make that transfer of materials in and out of the cell happen more efficiently. To tie this back into our different cell types, like I just said, eukaryotic cells are much bigger than prokaryotic cells, which means they actually have a much lower surface area to volume ratio. So the diffusion happens much more slowly and they should be less efficient. One of the questions in your lab asked you to think about how they can still function well, even with a slower rate of diffusion. And the answer comes if you think about the structures that eukaryotic cells have that prokaryotes don't. In addition to the membrane around the outside of a eukaryotic cells also have many, many membrane-bound organelles. So we are going to start shifting now into talking about all these different organelles and structures within eukaryotic cells. Your task today is going to be to go on a scavenger hunt around your house. If it's nice, you can go outside and get some things from outside as well. That's fine. You're going to be doing an activity to review the functions of a bunch of organelles that you learned in 10th grade, but we're going to review them quick today so that we can go a little further with this tomorrow. This activity is attached to today's classroom post. You may do this on your own, or you may complete this with a partner if you are able to truly work together. So if you can FaceTime each other or message back and forth or share the work that you're doing. Because the point of this is for every single one of you to review the functions of all 14 organelles that are listed on that document. If you are working with a partner, you only need to submit one assignment. However you decide to do this, please don't duplicate your information. You could either turn in, like if you divide up the work and one person does half the organelles and the other person does the other half, that's fine. But then only fill out half of your doc and make a note that so-and-so did the other half. The thing I want to avoid is I don't want to have to grade the identical thing twice. Or you can have one person typing on the document and the other person collecting the items or something like that. It is important, like I said, that you all are familiar with all the organelles. So if you do divide them up between you and a partner, make sure at some point you talk to each other and share what you did. And you can teach your partner about the organelles you did, and they can teach you about theirs. So make sure that you are getting that review of all of these. The first thing you need to do is look up the functions of these organelles and fill that in on the Google Doc. And then you're going to find items, like I said, in your house or outside of your house that represent the function of each organelle. Not what they look like, but the function and the job of what each one does in a cell. You can be pretty creative here. You'll need to describe your items on the Google Doc and kind of why you selected them. You also need to submit 2 pictures. One of them is going to be a picture of all the items that you find that represent all of the organelles within a eukaryotic cell. And then the last step that you'll see on the Google Doc asks you to go through out of all your items and select which ones would also be found in a prokaryotic cell and take a picture just of those organelles. That'll be a smaller group. So you'll be submitting 2 pictures, one of eukaryotic parts and one of prokaryotic parts. This assignment is due by Wednesday night, but I would strongly recommend, since it is the last week of the trimester, that you really try and do this afternoon and get this done today. so that you can just continue working on the other labs and assignments you have due throughout the rest of this week. Let me know if you have any questions. Have fun and have a great afternoon.